welcome everyone to the fourth session of our AMR webinar series. It's so good to see you here. Um, I will hand over to our moderator, Mr. Junior Shao. Junior, over to you.
Thank you, Yamanya. So, good morning, good afternoon, and good evening to everyone joining us from different parts of the world. On behalf of the organizing team, I would like to warmly welcome you to the fourth AMR webinar series, whose focus today is innovations and technology in combating AMR in Africa and other low and middle income countries. My name is Junior Alfishau, and I will be the moderator for today's session. As previously mentioned, our objective today is to explore how new technologies, ranging from rapid diagnostic tools to the use of artificial intelligence and big data in AMR surveillance, novel antimicrobials, and alternative therapies can accelerate progress against AMR, particularly in Africa and other low and middle income settings. Before we begin, just a few quick housekeeping notes. You'll see polls appear at different points in the session. Please do take a moment to respond, as your input will shape and enrich our discussions today. If you have any questions for our speakers, kindly type them in the chat and the speakers will respond. I will also select a few questions for the Q&A segment at the end of the sessions. Finally, please note that the session is being recorded for future reference. Today, we are honored to be joined by a distinguished group of speakers and panelists who bring expertise and practical experience in this field. I will introduce them to you shortly. For now, thank you for being here and for contributing to what promises to be an engaging and insightful session. Thank you once again for joining us, and let us get started. So please take about 2 to 3 minutes to respond to the poll, and then we'll summarize the poll results. As we continue voting, I will explain the session structure. So we'll have a panel, we'll have a keynote presentation, about 15 minutes, and then we'll have the in-between polls, and then we'll have a panel discussion. So from the voting that has been going on, we see a representation, a diverse audience representing clinical healthcare worker, research, academia, public health, government, industry, biotech. This diversity actually reflects the very essence of today's conversation, that addressing AMR requires innovation, collaboration, and perspectives from across sectors. So thank you for that. So as we move to our keynote presentation, so the topic is exploring innovations and technologies that can accelerate progress in combating AMR. Let me just share with you the bios. For presenters, we have 2 presenters for this session. So our first keynote speaker is Carol Raffel. Ms. Carol serves as the head of the Joint Office for the Southern Africa at the Global Antibiotic Research and Development Partnership, GADP, and brings extensive experience in global public health, particularly in HIV/AIDS and TB arena. She has over 20 years of leadership in both a pharmaceutical industry and community pharmacy. She has worked with organizations such as the J&J, Johnson & Johnson, GSK, Adcock Ingram. Her expertise combines deep knowledge of public health systems with valuable insight into the Southern African health landscape. Carol holds a Bachelor of Pharmacy from Rhodes University and a Master's in Public Health from the University of Liverpool, with additional training in business project management. We are delighted to have her with us today. to set the stage for a discussion on innovation and technology in combating AMR. Our next speaker is Dr. Eloise Dutoit. She is an antimicrobial resistance consultant and microbiologist with more than a decade of experience across academia, public health, and global health policy. She holds a PhD in molecular and cell biology from the University of Cape Town and is currently completing her master's in public health. Her expertise spans AMR surveillance, antimicrobial stewardship, microbiome research, and the development of national and regional AMR strategies. Eloise has advised governments and international organizations, including JH-PAIGO, the Global Antibiotic Resistant Partnership, the Global Antibiotic Research and Development Partnership, and the Botswana University of Pennsylvania Partnership, where she has contributed to policy development, laboratory strengthening, and One Health initiatives across Africa. We are very pleased to have our insights on today's keynote. Carol and Dr. Eloise, whenever you are ready, the floor is yours. Over to you.
Thank you very much, Junior. And yes, good evening, good afternoon, good morning to everybody, and a very warm welcome. Thank you to React Africa for inviting Eloise and myself to join you this afternoon. So just to let's get going, because I know we've got a bit to get through. I think someone's going to help me advance the slides. So if we can move to next slides, that would be great. Great. So we're going to cover these three themes in high level this afternoon or this evening, depending on where you are. We're going to talk a little bit about rapid diagnostic tools and some of the advances being made, a little bit about AMR surveillance, particularly touch on the role of artificial intelligence, big data, machine learning, and then round up with talking about the developing new antimicrobials and alternatives that are coming through in the landscape. Next slide, please. Before I get into the detail, I just want to make a bit of a disclaimer that both myself and then Eloise, as per the introduction, we work for an organization called GARDP Africa, which is part of the GARDP Global Network, GARDP standing for the Global Antibiotic Research and Development Partnership. So really what this organization does is really the focus on that third topic, which is about developing new antimicrobials, antibiotics. So we're not per se experts in rapid diagnostic technology or big data and surveillance, but certainly we are, within the ecosystem and have tried to pick a few examples for you this afternoon on all of these themes to say what is going on. So we'll start first with the rapid diagnostics, and I will hand over to Eloise just to take us through this section, and then I'll pick up after her on the last two topics. And I think there was just a comment to try and put the slides onto slideshow. So I'm not sure if somebody can do that while we move on. Next slide, please, and over to Eloise.
Hi, good afternoon, everyone. Thanks very much for the introduction. My name is Eloise, and thanks so much for the invite to share some thoughts on the area of rapid diagnostics in the field of AMR. Next slide, please. So the development of new rapid diagnostics would optimize treatment in general. And the image on the right that you can see is a scenario where a rapid diagnostic leads to a doctor's visit where instead of the doctor looking down your ear or throat to look for clinical symptoms of illness and starting you on empiric therapy, she instead has a rapid diagnostic test that can tell you within minutes if you have an infection and if it's viral or bacterial. If it is bacterial, she can then prescribe you the best line of treatment. Next slide, please. Thank you. So even though that you can go back. Thank you. Even though we know that there's a desperate need for diagnostics in the management of AMR, the investment into diagnostic development has remained extremely low. And you can see this by the yellow bars over the years from 2017 to 2019. And if we thought that investment for new antibiotics was low, the development of new diagnostics is even lower. Next slide, please. So the Find AMR Diagnostic Test Directory, which is a very useful platform, shows that there are many tests. Some are already in use and some are under development, but there are gaps remaining, and some and the sustainability of their implementation is always questionable, and especially in LMICs, where a recent report has shown that 1.3% of labs in sub-Saharan Africa carry out routine bacteriology culture. In addition, 28.5% of the rural labs in sub-Saharan Africa have access to electricity. You can click for the next slide. Thank you. So the ideal point of care test would be affordable, sensitive, specific, user-friendly, rapid, equipment-free, and be deliverable. It would also answer 4 questions. It would answer if your infection is bacterial or viral. If it is bacterial, what are its resistance mechanisms? If it is susceptible, which is often a more confident piece of information than resistance mechanisms. Thank you. Next slide. So in general, we have 3 different groups of diagnostics. We have the culture-based assays, which give you pathogen identification, and that can be through VITEC or multi-top, for example. You can then proceed to antimicrobial susceptibility testing through disk diffusion or VITEC, for example. We then move on to the lateral flow assays, and that can give you pathogen identification. And a very good example of that is the CRAG, the cryptococcal meningitis antigen detection. You can also get the resistance mechanisms identification, and for example, the carbapenemase enzymes, as well as biomarkers like CRP or PCT. When we move to molecular assays, we have things like PCR, gene expert, next generation sequencing, and whole genome sequencing. Next slide. So when it comes to culture-based, it is the current gold standard, and when reporting to the GLASS surveillance system, it is a requirement, culture-based identification and sensitivity. However, it is time-consuming, and given the limited lab capacity in LMICs, it is clear that this is an area for development. Lateral flow assays are expensive. The procalcitonin biomarker lateral flow assay is extremely expensive and often not available in LMICs. Again, molecular assays are costly, there are limited targets, and their efficacy, especially in LMICs, needs further assessment. So what would we like to do currently and moving forward is, with regards to culture-based assays, we need to improve lab capacity in LMICs. We can use technology like Antimicrobial, which I'm going to speak to shortly. But the good news is that technology such as affordable and reduced size systems for blood cultures will be coming to low and middle income countries soon. With lateral flow assays, we would like to expand the number of target pathogens, resistance mechanisms, and biomarkers. We would like to improve the clinical relevance and specificity of tests such as CRP. We would also like to include new biomarkers to differentiate between bacterial and viral. And of course, if we could make this a culture-independent method, that would save time. When it comes to the molecular assay, very similar things. We just need to further assess the efficacy, and then culture-independent assays would be helpful too. Next slide. So although I've shown that there are some rapid diagnostics available, not all of them are applicable to LMICs. And so this just really makes us think that we need to be innovative and we need to start thinking out of the box. And a very good example is the use of cane rats in some parts of Africa that were used to sniff TB out of sputum samples. I'm not saying that this is the most reliable test, but it's a very good example and shows the way for further research into the use of e-nose devices for diagnosis. Then I would just like to highlight the winner of the Disruptive Technology Award system. The one on the left from the winner of 2023 uses single cell biology, where they encapsulate every single cell in a polymicrobial mixture. And in that little capsule, they will carry out the susceptibility testing and report that. So you will get your identification of the bacteria as well as its susceptibility. And then one of the finalists in 2025 was this cassette where you put your sample into the cassette and 20 minutes later, you have your ID and susceptibility. Unfortunately, in LMICs, neither of these are affordable options. Next slide. So moving on to Antimicrobial, which is a MSF and World Health Organization innovation. It is a mobile AI-powered tool developed open source. It's validated in resource-limited settings, and it's currently being rolled out in several African countries. It is designed to make accurate interpretation of disc diffusion AST plates, and it then recommends the right antibiotics. It also then automatically feeds into data into national and international surveillance systems. Next slide, please. So it does this through an image analysis program. It first of all takes a photo of your AST plate. It then recognizes the antibiotic disc as well as the inhibition zone. It then uses AI to apply expert rules, and it makes antibiotic recommendations, which are sent to the clinician. And it also recommends additional tests and raises IPC flags. So it also detects resistance mechanisms and uncommon phenotypes, as well as incompatible results. Next slide, please. So Antimicrobial is great in that it addresses quite a few gaps in future directions. It is used for accurate susceptibility interpretation, where trained microbiologists are scarce. It supports antimicrobial stewardship by reducing misinterpretation. And future directions include its integration with electronic lab systems and expansion to more pathogens and broader LMIC rollout. Thank you.
Great, thanks very much. I'll move on quickly now to talk about AMR surveillance and big data. So next slide. And I thought what I would do, with permission, I have shared a few slides because this was actually a topic that was presented at the G20 at the Health Working Group a couple of months ago in the AMR session. So sharing a couple of slides here with permission from Chris Kelly, who's a data scientist here at UCT and working in this space. So literally, this is a copy and paste of the slides that was before the G20, where as we really move into the future and all of us holding hands, if you like, to face the growing threat of antimicrobial resistance, we have to start thinking about embracing this new paradigm of using real-time integrated intelligence surveillance systems. I won't read through all of that slide, but I think some important points is that although some of these systems are starting up in human health, it's really critical to start thinking about this type of surveillance across the One Health continuum, and think about how we can promote and indeed implement data sharing, which we know is always a difficult point, but really think about how these big data systems can be shared. Next slide, please. So I wanted to highlight CamoNet, which may be familiar to some of you because this is an initiative that is operational on the continent, and you'll see there in the map that there are some national hubs in Uganda and South Africa and technical support systems in Malawi and Ghana. And you will find that they have a very active website. So if you don't already know, I'd encourage you to go onto the website and understand how they're developing out the networks and the research really behind CamoNet. And the next couple of slides I'll show you, next slide, is really some of the work that Chris is doing with CamoNet in the Western Cape here, where Eloise and I are, to start exploring some of the systems. So I don't expect you to make sense of this slide. It is, I think, for most of us that come from a healthcare setting, quite overwhelming to start thinking about big data. But effectively, you've got all of those little blue data sources coming in on the left-hand side, and then those middle two columns is really where the machine learning and the big data scientists kick into programming and working, and it really extracts all this data, packages the data, and then spits out on the right-hand side different sets of data that would be useful for different audiences. So be it the patient, be it for clinicians, be it for facilities, community providers, administration, and really the list is endless in terms of which are the users that can then extract and start to think about how to use this big data for decision making. And the next slide is really just an example of the type of dashboards that can come out of the data and out of huge amounts of data inputs. Now, obviously, this one is designed for a particular audience. And again, I'm not expecting you to make sense of this. It's a very busy slide, but these are the kind of dashboards that they are trying to work on to be able to, design data that would then be useful depending on the audience that this particular, the questions that they're asking. Here you'll see that it's about carbapenem-resistant organisms coming out of the hospital groups in the Western Cape. But the next slide really just summarizes it in a very simplistic form. That yes, on the left-hand side you have the big data coming into some kind of AMR dashboard, similar to the one that I've shown you in the previous slide. And that can feed clinicians, pharmacists, ID specialists, and so forth. But I think to think even bigger is the kind of depiction on the right-hand side, and I apologize for the size of the slide, but I think you can get the point, is that it's also really important that this big data is then able to be sliced and diced, if you like, to be able to inform research and what do new drugs need to look like. Importantly, how to think about some of the social science messaging. What is the data telling us about the messages that need to go out for public awareness, or more creative messaging around how to combat AMR. So obviously critical that it's robust and evidence-based data. Moving on to the last of the topics and the development of new treatments and alternatives. Next slide. I very quickly just want to, and I know you're not going to read these slides, but really just try to tell the story, is that, as I think many of you know, there are fewer and fewer classes of antibiotics that it's really slowed down. The pipe has slowed down. You've got major antibiotic developers that have exited the market or continue to struggle. We know that just the money, the venture capital, etc., is not going into antibiotic discovery and being diverted into more lucrative, if you like, healthcare areas. So I think it's really just important to remind us that although the research and innovation can go on, it's really important about how do they come to market. This is not an easy marketplace. And just using the example that I think many of us know with antibiotics is that there may be a handful of new antibiotics that have been developed, if you like, but still not really coming to market. So although I'll talk about the innovations and the technologies, I just want you to keep in mind that we also need to crack, if you like, the mechanisms by which these innovations do come to market, particularly in resource-limited settings where we know there are even greater barriers to come to market. So with that, next slide. I'll talk about three. Then I was just going to say, so it's one of the reasons why GuardP, which is a nonprofit product development partnership, was created, is certainly we can't solve all of these problems, but being able to try and not only encourage the pipeline to be developed, but also to bring these innovations to market. So next slide. I'll talk about three areas of antibiotic or new technology coming to market. The first is something that I think we're more traditionally aware, and can we get new classes of antibiotics into development? So I just wanted to highlight one recent example, and nobody can say that word, so I'm not going to try in terms of the class that's come along. And this has got a novel mechanism of action that is distinct from fluoroquinolones, and the first antibiotic in class to really be developed is zoliflodacin. So I can speak to this with a little bit more more insight, because it is an antibiotic that GOD-P has developed in collaboration with IST, an American innovation company. So it has been designed to tackle gonorrhea, Neisseria gonorrhea, and really there hasn't been a new antibiotic for gonorrhea in decades. And just why this has been such an important pathogen to tackle is that ceftriaxone-resistant Neisseria gonorrhea is a top priority pathogen. Ceftriaxone, which is largely what I think many of our countries are using as the standard of care, is the last line to treat Neisseria gonorrhea. So at some stage, when resistance rises, we really need something in the pipeline. So this is currently an antibiotic that is before the FDA for approval, and hopefully we'll start to see further sort of commercialization of this antibiotic in the coming months and years. So, you know, as the ceftriaxone resistance, which is inevitable, there is a backup. Next slide. But behind some of these new classes of antibiotics is really about what can AI do. And I'm sure you would have seen recently in the press more and more discussion about AI and drug discovery. So I just wanted to draw our attention that indeed on the continent, through the University of Cape Town and the translational AMR research called STAR or CETAR, there has been a recent investment into Kelly Chibali's laboratory, and really being putting money and expertise and resources into the drug discovery development here in Cape Town that can start to tackle antimicrobial resistance. So good news. And as I think many of you know, his foundation reaches into many of the African countries trying to create a drug discovery network for the continent. Next slide. But of course, behind this drug discovery, as I mentioned, is really about the AI and the power that AI can bring to discovery of new antibiotics, because it really speeds up the process of seeking candidates or hits for the discovery process. I think as of last week, there was quite a bit of media coverage around some new work that was done in the States, but nevertheless, I think just demonstrates the power of AI. And again, particularly, they were looking at trying to find new hits for gonorrhea. And an important link here for our continent, and again, a quote there that I've just used from Kelly Chibali, is that it's going to be really important to use AI in a sense to make sure that we're coming up with drug discovery that is well aligned to African needs. As we know, a lot of drug discovery has been done that it doesn't necessarily line up with the African genome and African descent. So it's going to be an important combination of using AI not only to speed up the process, but also to make sure that Africa can take a lead to make sure that those candidates coming out of drug discovery are going to be well suited to our ethnic profiles on the continent. Next slide. I'll touch on phage therapy because this is still, it's one of the new alternatives that is in the wings. It is complex. So it is not, it's not something that is imminent for us to sort of have in a commercial setting, but I think it is one of the new innovative approaches that are a bit closer than some of the other innovations, which are still quite far out and still very sort of academic, if I can put it that way. So just quickly, what is phage? What is a bacteriophage? And so really it's a virus that would specifically infect and kill a pathogen, a bacterial pathogen. And there's quite a lot of interesting dimensions to phage therapy because they are very specific. So they would only kill the target bacteria, and they would leave the rest of your microbiome, your beneficial bacteria, alone, unlike what we know our traditional antibiotics would do. It is, you know, as long as there's a target bacteria, so that pathogen of interest, the phage will continue to replicate inside of that bacteria and kill themselves, and then they will just die off once that target bacteria is under control. So they're very good toxicity profiles, and of course, in this context that we're talking about in the threat of AMR, they would be active against resistant bacteria. So next slide, please. And I just wanted to highlight two two pieces of work. So recently, a couple of months ago, there is a workshop report that you will find on the web. That was a meeting convened by the Global AMR Hub, including the relevance of bacteriophages in low resource limited settings. So if anybody is particularly interested in this area, I would refer you to that report. And then to say that we do have some phage research happening on the continent, and I'm sure there's more than just this one that I've picked. But here is a professor working in Guinea at the Institute Pasteur of Guinea, where they're really starting to try to develop phage cocktails within the African context. And as you'll see there, they hope to carry out first clinical trials within the next 5 years. But a complicated space, an unclear regulatory path about how phage therapies would be regulated, but certainly an interesting space to watch. And then to end off, I will touch on the last topic, which is probiotics. So again, a new innovation that is perhaps closer to the clinic than some of the other innovations that are a bit far out. And in particular, there's been some interest in the use of because of the use of antibiotics in preterm infants, which we know are often subjected to antibiotics because they're at risk when they're in hospitals and in neonatal ICU type settings. But the use of antibiotics is really altering, if you like, their antibiotic resistant genes. So it does get quite complicated, and actually Eloise is more of an expert on this than I am, so I'm not sure why I'm talking to this slide. But really the idea is that if you are able to use probiotics at the same time, it can help to reset these bacterial communities, the normal gut bacterial communities in these very young babies, and will actually slow down the spread of AMR. And there's a very recent paper, again, one that I think came out, if it wasn't last week, it was earlier or the end of last week. So again, you would be able to find this available, really breaking down the impacts of antibiotics and probiotics in these very young babies already around the world. So for instance, there's a statistic there in the UK that they are adopting this practice of using probiotics. And I think this presents a very simple and probably very cost-effective intervention that we really should be giving more thought to in our types of settings and in our particularly with our young birth weight babies. So with that, I hope we have kept to time. I know there was a lot to go through. We are a little bit over, but very, very, very grateful for your attention, and back to you, Junior, as the moderator.
Thank you, Carol and Dr. Lois, for your wonderful presentation. You have clearly highlighted three critical fronts in the fight against AMR. You touched on diagnostics. So it's true we need faster, cheaper, but simple tools that will work in all settings to ensure the right antibiotic is used at the right time for the right patient. You've touched on surveillance and AI and big data. We see them as game changers, enabling real-time tracking of resistant patterns for smarter and faster responses, even in resource-limited areas. You've spoken of new treatments, so the pipeline is not dry. From novel antibiotics that are being discovered by AI to promising alternatives like phage therapy as well as probiotics. So we can agree that innovation is proving or is providing new hope in the fight against AMR. So I think this sets the perfect stage for our panel, who will dive deeper into the practical implementation of this innovation. So just before the panel, we can see we have a poll session. Kindly take time 2 to 3 minutes and respond, or I see the results. So it was clearly asking on the awareness on AMR diagnostics or innovations. We see 77% aware of rapid diagnostics, that's the point of care testing. AI, about 17%, so it is still a new field, AI and big data. New antimicrobials or alternatives are still, we are getting there step by step, and we still have a small percentage that are not familiar. So with this said, I will introduce our panel shortly.
Maybe you can
Allow
take
me
one or two
to
questions
share.
for, maybe you can take one or two questions for Eloise and Carol. I can see there's a hand up and there's a question as well in the chat.
All right. Thanks for the reminder. Cyprian, can you please unmute yourself and shoot your question? Then we'll go to Papi. Over to you, Cyprian Baribio. Hello, Cyprian. Please unmute yourself.
Yes, thank you. I just want to say that it was just an error.
All right, all right. Thanks for that.
Thank you.
So there is, maybe we'll take the question in the chat box. What is the future of bacteriophage therapy in Africa? Over to you, Dr. Eloise and Carol.
Sorry, just trying to get myself off mute. Yeah, I mean, I think it's I certainly just want to say, first of all, I'm not an expert in this space. I think it is one of those innovations that are probably closer to markets than some of the other innovations, but it's still a long way off. As I say, there are many challenges in the idea about, how would we get these registered, cost, which I think is always going to be a constraint. But I think it definitely holds future in the sense that we're not going to be able to keep developing new classes of antibiotics and just stay ahead of the resistance curve. So we have to be thinking about different innovations that are going to work against resistant bacteria. And there's going to be some that are just going to outpace us in terms of antibiotics. So I think it's technically complex. And I would say, I don't know how many webinars we're going to have under React Africa, but perhaps even approaching the gentleman that I had in the slide there, who is now running bacteriophage research on the continent, and getting his perspective on how he thinks this might fit, might be a really good opportunity for us to understand how the phage therapies are going to fit. I can say I don't think we're going to be seeing him in the next 5 years, but maybe we may see some clinical trials on the continent in the next 5 years. And I think that will start to answer some of the very practical implications. Can we register, and can we work against the critical pathogens that the continent is seeing? And can we see a day where we could administer them to patients? But, being very safe, and being able to work against resistant bacteria have to be criteria that makes it very interesting for the continent. Thanks, Junior.
Yeah, thanks, Karen. Menyanga, hopefully it's been answered. We move to Papi. Papi, can you unmute and ask your question?
Merci beaucoup pour la parole. Je m'appelle Patrick Chika. Je suis microbiologiste.
Ichika, I'm a microbiologist from the Democratic Republic of Congo. I'm not sure. Are you able to hear me? Are you able to hear me? Can you please confirm?
Yes, we can hear you.
Yes, we can hear you.
Oui.
Est-ce que,
I would like to know.
voilà, la question était de savoir, est-ce que les tests de diagnostic rapide pour détecter la résistance aux antimicrobiens, pour détecter les germes,
rapid diagnostic test.
je ne sais pas comment ça fonctionne.
I'm not sure. How does that function? Is it from the sample? Should you take the blood, or we have first to cultivate the sample to incubate that to have the colony of bacteria, and maybe yes, you are now going to use the bacteria to detect the resistant bacteria. You know that you've talked about. So this is the first question. Second question: how do you make available these test rapid test? If we talk about the prevalence of the bacteria, so you have a resistance of 10% in Lubumbashi in the DRC. Now we find that it's too big, but it's a challenge, that we are all facing. But we have to know the availability. How is it the availability of these rapid diagnostic tests? Thanks. These are the two questions that I had to pose or to ask. Thank you so much.
Thank you, Papi. Dr. Luis.
Hi, Junior. Although I speak quite good French, I couldn't catch all of that. I'm not sure if there's a translation available.
Yes, there is.
Yes, the interpreter is available. Were you able to hear the interpreter?
Is it going to pop up, or do I need to find it somewhere?
It should be at the bottom of the screen.
Oh, I think, am I still looking at your screen?
Perhaps you could just read it out.
There is an icon. There is an icon like a globe. I don't know if you are able to see that.
Sorry, we're not sure where to see the translation on our side, so
I think the translator is in. Yamanya, can you ask him to interpret that?
Je peux poser la question directement en anglais alors. Okay, I said that here in Congo, we need those kind of tests because right now I'm watching or I'm following the presentation of graduation for a pharmacist who has presented for he's presenting us that the prevalence of extended spectrum Enterobacteria producing from urine samples. She has presented that she found a ring, a tin for prevalence. We can see that that's the more important challenge that we have got in our country or our continent of Africa. So my question was to know those kind of rapid tests are available. If they are available, how we can get it in our country, DRC? That's the first question. The second one is to know how to use the kind of rapid tests. Is it through the sample, for example, urine or blood? We can take that blood or urine and directly we can use it on that rapid test, or we have obliged to cultivate the urine first to get colonias after 24 hours incubation, and then we are going to use the colonias on that rapid test. Those are my two questions that I would like to know. Thank you.
Thank you, Papi.
Thank you for the question, Papi. And if I don't answer your question, please do feel free to contact me later. I couldn't hear everything in full detail. So the diagnostic tests are available commercially, depending on which test you'd like to do. So the ones that are available, you can contact the suppliers, and they will be able to supply those tests to you. You just need to confirm, first of all, if that test is currently available. Yes, there are tests that you can use urine straight. You can use the specimen of urine straight onto the rapid diagnostic, and other lateral flow assays. Some require bacterial culture before, and then you use pure culture onto that lateral flow assay. But others have been developed so that you can use it straight from blood, straight from spinal fluid, straight from sputum. So using it from a variety of specimens is possible.
Yeah, thank you,
Okay,
Dr.
thank you
Eloise.
for your response. That you can share with us your email or your address to contact you.
So we'll pick two more questions, then we'll move to the panel discussion. There's a question in chat here talking about One Health approach in terms of AMR. Are there any effective measures to involve all stakeholders in combating these AMR issues? So you can, either of you can pick that one up. And then we have, I think, there's a question on the new antibiotic, if it is available. Let me just check where it is.
Dorothy, while you find that, Junior, I'll say on the One Health approach, I think obviously it's an extremely broad space, and I think just to stress what I said in the actual presentation, that being able to start to gather data from all three of our sectors, environmental, animal, and human health. I think a lot of the complexity of being able to work across all of these three sectors is going to be aided or sort of reduced, if you like, if we're starting to collect data and can put it into these big data systems. Now, I think it is going to take investment into technology platforms so that and obviously we know that if you don't put good quality data in, you're not going to get good quality outputs. So this is indeed a challenge. But I think once we are able to start collecting data from all three of these sectors, and we're able to start to see whether there are cross-cutting themes that will then be able to guide actions. At the moment, I think what we tend to do, because we're sort of forced to do, is to look at the three different sectors somewhat in isolation. And yes, we know we've got themes, for instance, how do you reduce antibiotic use in animals, but trying to link together what these impacts are going to be, I think, is incredibly difficult. And this is hopefully where AI, machine learning, big data will be able to start to make some breakthroughs. And I think even if there are areas, to have it across the continent all in one go, I know we're a long way from there, but even if there are areas where they are able to look at, input from three sectors, there will be some learnings that we can then scale to other settings, other countries. So at the moment, are there one intervention that we can use across all three spaces, animals, human, and environment? I can't think of one off the top of my head. Obviously, we don't really use antibiotics in the environmental setting, but we know that we put environmental waste into the environment that are antibiotics. So yes, antibiotic usage and waste of those antibiotics, how we manage the waste is maybe one theme. But I think this is where big data is going to be able to leapfrog us into a different type of thinking, where we can link these three compartments together.
Thanks, Carol. I think in the interest of time, we'll move to the panel discussion. So I think the rest of the unanswered questions will get back to you in the chat box. Let me introduce our panelists. So we have Ms. Olabisi Lawal. She's a medical laboratory scientist and researcher specializing in antimicrobial resistance. She's a graduate of the University of Benin in Nigeria and brings variable expertise in clinical settings and laboratory-based research. Her work focuses on advancing our understanding of resistant pathogens and their impact on global health, making her perspective highly relevant to today's discussion. She will provide insights on rapid diagnostics and early detection. So our other panelist is Dr. Kiplangat Sigei. He's a medical affairs manager for the Anglophone Africa at Biomuro, which is a global leader in vitro diagnostics. In his role, he leads scientific and medical engagement across East, West, and Southern Africa, supporting the company's clinical diagnostic portfolio. Dr. Sigei has been an active member for Kenya National Antimicrobial Stewardship Interagency Committee since 2019 and contributed to the development of Kenya's first national antimicrobial stewardship guidelines, which were launched in 2020. He has also served as a co-investigator in the global PPS Kenya study in 2021, which provided critical insights into antimicrobial consumption and resistant patterns in hospitals. He holds a medical degree from the University of Nairobi and a master's in public health from the London School of Hygiene and Tropical Medicine. We look forward to drawing on his experience on new antimicrobials and alternative therapy, lessons learned, and future directions. So with our panel to set the stage, I will go straight to questions. So our first question is directed to Ms. Lawal. Please, other speakers, please feel free to chime in as she answers this. So the question is rapid diagnostics and early detection. So what barriers, be it technical, regulatory, or financial, exist in deploying point-of-care diagnostics at scale in resource-limited settings, and how have successful programs overcome them? Over to you, Olabisi, when you're ready.
Okay, thank you so much. Hi, everyone. It's great to be here today. So, our question, I personally think when it comes to rapid and accurate diagnosis, there are lots of barriers, especially in limited resources countries like Nigeria. So, speaking of technical barriers, many diagnostic tools, they need, things like stable electricity, cold storage, and regularized environments. So these are very, very important when it comes to, using rapid diagnostics like POCT. So, and many of these things are not readily available in primary health centers, rural environments like in Nigeria here. So there is often a storage, even trained staffs, they are not, very efficient in using most of these tools correctly. So this can also make it harder when it comes to getting the best out of most of these POCT testing tools. And another challenge in technical barriers, I think, is, the test might not really fit into the doctor's day-to-day activities. And if the doctors do not trust the result that's going to come out of the test kit, it's most likely that they won't order for the test. And also the turnaround time, if it's taking too long, so the test might not really be useful. For example, over here in Nigeria, many local facilities are now using malaria outbreak, the rapid diagnostic test. So, and these tests were designed, they were designed to work in different types of environments. they are heat stable, they are actually very cheap to use. So, and even community healthcare workers that are not really, they're not specialized, they're not medical laboratory scientists, they can also make use of this diagnostics and this POCT testing. So I think malaria RDT has actually overcome that, and they were able to produce these kits to be used in this kind of settings. Another one is the gene experts. Gene experts too also requires, minimal experts when it comes to using it. So this has helped to improve the detection of TB in limited resources limited areas like especially northern Nigeria, where it's very difficult to come by, specialized scientists. So the other barrier is the regulatory barriers. Regulatory barriers, there are lots of regulatory process in many resources limited countries. And this can make things slow and makes them more fragmented. So diagnostics often needs approval from different agencies, and this can cause delay and even increase the cost of these POCTs. So on top of that, there's a lack of unified quality standard, which means that sometimes this low quality test can also cause mistrust. So misaligned timelines and between regulatory approvals, getting approval and procurement process, it can also create gaps in getting these POCTs, like having access to it. So for example, when COVID-19 hit Nigeria, the NAFDAC, that's the National Agency for Food and Drug Administration Control in Nigeria, they played a key role in getting and fast-tracking approval of rapid antigen testing for most centers. So this also helped in quicker regulatory processes. And there were a lot of testing that were being done, especially in high-risk locations as at that time. So this agility actually helped in controlling the regulatory bodies, and they were able to respond effectively when there is political will. So I hope that looking ahead, the African Medical Agency will be crucial in harmonizing approvals from continents to make it easier to roll out, diagnostics testing faster. And another barrier is financial barrier. When it comes to financial barrier, I think is the major barrier faced by most LMIC. The cost of using all these sophisticated diagnostic testing, they are very expensive. And most organizations cannot even afford it. So most of these organizations, they have to, mostly rely on funding. And when it comes to funding, sometimes it might not be sustainable. So, the barrier of funding can actually be, we can tackle it by proper reimbursement structures might be put in place. Private companies, can invest more in diagnostics, and this can also help in having easy access to all these POCTs. For example, in Gene Experts program in Nigeria is actually a funded program. It was mostly funded through food procurement, and it made the test easier. Like people were able to access the testing. Even people without money, they can easily have the tests and get the results. So by teaming up with international partners, Nigeria were able to bring the price of TB diagnosis down and making it more affordable for patients. Same thing with the malaria disease now, especially in rural areas in Nigeria, like in Borno, stuff like that. So for antimicrobial resistance, we can learn from all these lessons. So to tackle AMR, I think we should invest more in affordable and portable POCTs that can detect resistance markers, just like malaria RDT. I'm pushing for stronger regional cooperation and faster regulatory approval, much like the AMR, that's African Medicine Emergency, just like what they are doing, and exploring financial options like food procurement to lower the cost of advanced diagnostics will be really helpful in tackling antimicrobial resistance in LMIC. So I think scaling rapid diagnosis for AMR is not just about technology. I believe it's more about creating the right systems. And when we combine innovative things that seems to be possible, just the way we've been able to do with malaria and TB, and we can also do for antimicrobial resistance too. Thank you.
Thank you, Olabisi. Anyone from the speakers who wants to add in? Or maybe I can start. Perhaps we can think of technical barriers like we'd say most of the remote areas do not have electricity. It could be a barrier as well. Lack of technical personnel could be another barrier. I think those as well contribute to why scaling up rapid diagnostic to remote settings is proving a problem at the moment. Kiplangat, do you have any addition on that? Dr. Sigay?
Yeah, thanks, Junior, and excellent overview, Ms. Lawal. I think the barriers have been very much highlighted, and I think beyond that, I think one thing to add would be obviously some of the rapid diagnostics do have their own inherent challenges around sensitivity and in some cases specificity. So for me, if I may, instead of talking more about barriers, I could actually share a solution. One of the things that we should also consider when we try to address the issue of diagnostics is diagnostic networks. And what I mean by diagnostic networks, for those who may not be familiar, is really maximizing what we already have in our various countries or institutions. One of the speakers mentioned Vitec solution, which is actually from bioMérieux. This is not a solution you can have in every hospital, regardless of how much funding or technical expertise we have. So sometimes you can actually have a diagnostic network where a hospital can serve as a reference or a referral to receive samples. And if I may, instead of just looking at rapid diagnostics, we can actually say rapid diagnosis, because sometimes a rapid diagnostic tool, of course, is very important and we need it, but not all results will come from a rapid solution. So you could then refer samples, especially for confirmation, to centers that have higher level diagnostics. And at the end of the day, people want results, whether they have a rapid test or whether they have the most advanced solution. What we can have is a diagnostic network that enables results to circulate as they are needed. So that's probably what I would add to this. Thank you.
Thank you for that. So we'll move to our second question. I'll ask Dr. Luis as well as Carol to share insight on this. It's about AI and big data and AMR surveillance. The question is, how can artificial intelligence and big data analytics strengthen AMR surveillance in Africa, given challenges like fragmented health data systems and limited laboratory networks? Over to you, Dr. Luis.
Yeah, so, I think obviously the silver bullet, if you like, is to have very sophisticated AI or electronic systems, and we know that is it's not going to be feasible in all settings all the time. But I think I'd like to come back to the example that Eloise was presented about the Antimicrobial, because this is an example of using AI to help surveillance that is very kind of user-friendly. As a design that's come out of MSF, they really are trying to think of ways to bring technology that is low tech, if I can put it that way. It can be used offline. It can be used in settings where you don't necessarily have sophisticated data systems. So to just reflect on this a bit, I mean, it obviously you need a cell phone, but to be able to take that picture of that plate, and then AI and machine learning is really then doing the rest of the work and being able to interpret what you're seeing on the plates, and spit out an answer, if I could put it that way. So yes, the clinician is getting a real-time machine learning answer. But then if you are connected or can later connect to Wi-Fi, you then can hit a send button in the platform, and then that feeds into the big data, the surveillance system. So it's the same result that would feed into big data and surveillance, and obviously that depends on the strength of the electronic system that is in the surveillance, but also gives the clinician a real-time answer. And I think the more data that gets fed into the system. So if I could put it in simple terms in a way, the more photographs that get taken of plates, the more the machine will start to learn itself. And so it will recognize more bugs, it will recognize more AST zones, and it will start to improve. It's sort of a crowdsourcing kind of thing. So I think these are the type of innovations that are going to be really interesting for low resource settings. And I think this is where we have to start. I think we get a bit overwhelmed when we look at these big data systems and the type of infrastructure that everybody needs. It's like, well, we're never going to have that. Now, it'll take time, maybe we'll get there, but in the meantime, in the short term, these type of really clever systems like Antimicrobial, and I know it's not yet rolling out, I think it's still being used within MSF clinics, but again, I'd encourage you to go to that website. I put it on the slide there to start exploring, how it works, and obviously as it starts to roll out, the more people that can use it, the stronger the system gets. So I hope that gives us a little bit of hope and not completely overwhelmed by these very sophisticated electronic systems, and feeds into surveillance systems as well, and hopefully more accurately into the surveillance systems, because it's a machine reading the plate as opposed to a human who may bring error depending on how skilled they are. Thank you for posting that in the chat, Manya.
Thank you, Carol. So I was thinking of supercomputers when you hear big data and everything, but simply a phone can help us to have this data analyzed. Dr. Eloise, do you have something you want to share on the same topic, AI and big data?
Yeah, so no, just what Carol said about the importance of whatever kind of rapid diagnostic that is being used. If there is some way that the information and results can be fed into some sort of surveillance system, and preferably automatically. So, a lot of the rapid diagnostics that I spoke about, which were not feasible in low to middle income countries, were automatically linked to feeding into a surveillance system. Obviously, in lower middle income countries where perhaps we're using something like a lateral flow assay, that would have to be manually input. So yeah, that's it from me.
Yes, another key point you've mentioned, manual entry, duplication of work in the lab. So I think we can move to the next question. This is directed to Dr. Sigei, new antimicrobials and alternatives, but the rest of the team can still chime in. The question is, how can partnerships between academia, the universities, industries, and government accelerate the development and equitable distribution of these novel interventions? Over to you, Dr. Sigei.
Yeah, thank you so much for the question. Obviously, this tripartite collaborations between academia, industry, and government are really excellent in an ideal world, whereby academia serves as the research engine, industry serves as the commercialization and rolling out engine, and of course, government ideally supposed to fund and regulate and ensure that these are available. There are many examples. I think it's easier to share examples, and many in the audience will be familiar with them. Obviously, I'll start with GADP since they are online, and I think they're a better place to discuss that. Although they did give a highlight of how GADP works, I do believe there is partnerships there that cover the three entities. We have a few such as CABX, DNDI. There's one interesting one I saw from Australia, which is the AMR Hub, which prides itself as being one of the first in the world to do that kind of collaboration. So maybe bring it closer to Africa is usually the challenge I see with this. It is a perfect combination, but the reality is funding really keeps this kind of collaborations from shining. We have a lot of projects that are well funded, but as we have seen, funding models are changing. We've had some big shocks, especially this year, from US-driven funding. And other funding partners from other countries have also reprioritized or scaled down. So for this to work best, I would say in Africa, we need to find a way to fund it so that it really is functional. We depend a lot on funding from outside, which is okay because it has also helped to lead to these innovations being driven across our region. We've seen the new antibiotics that were shared by previous speakers. Without external funding, would probably not see them. But the issue is how do we then introduce funding? And yeah, so we've had good examples and they work quite well. We just need to see how to expand this beyond dependence on certain funders. And obviously, we need to ensure that ultimately the access to those who need it is most important. And so this is something that also is a gap that is still yet to be filled. There are models that look at tiered pricing, those that look at strengthening health systems and so on. Yeah, so I think that's my take on that. And I'll invite anyone else who wants to chime in on that.
Yes, thank you, CK, Carol, and Tim. Do you want to add anything on that?
The only thing I would say is that obviously there's never been a more important time to collaborate, and I think it's about focus as well and being able to find, and I'm talking very generally here. So I think it's really being able to align across different players. What are the I don't want to say two or three because there's always more than two or three, but in all being pulling in the same direction. So we know that there's a lot of ideas that are floating around, and I talk about from an access point of view. We all know that there's a dialogue around harmonizing regulatory efforts, about thinking about pooled procurement, and it's how do we make that happen as groups of Africans, right? And I think that's if there's anything, it's a sort of this call to action, this call to urgency. I really hope that we're not going to be sitting on a call like this in 5 years' time, still talking about pooled procurement, and it doesn't have to start with everything being pooled procured, for instance, or everything going through. I was really, really thrilled to see, I think it was last month, the Green Book, which is the sort of AMA, our harmonized regulatory. The Green Book has now put another, I think, 3 or 4. Can't remember exactly what they were. They weren't antibiotics, but adding to the Green Book. And I think it's really, if we can continue to make some of these initiatives work and show that the case, and start talking about what is being pool procured, what is being harmonized in a regulatory fashion. That's just on the access side. I think it's going to build our confidence. It's going to build our imagination to say that this is possible. So it's really, I think, a call for all of us to be able to rally around a few priorities and show that it can work. So we sort of move from talk and move to action. Easier said than done, but I think that's really what we need to do. We need to have some successes so that we can imagine how that can expand, because otherwise it's very overwhelming for all of us. And clearly the money is getting a harder and harder, it's harder and harder to think about how this is going to be funded. So we have to put some of these optimization tools into play.
Yeah, thank you for that insight. I think we should look into how we can leverage the regional as well as the regional platforms like AU, Africa CDC, and see how they can assist in especially the pooled procurement and the regulation part. I think in the interest of time, we can go to the next question. It's on implementation and policy. So I'll just shoot it out to anyone in the panel. What strategies can policymakers and health system leaders adapt to encourage adoption of new technologies and innovations while ensuring quality, affordability, and accessibility? I think we touched a bit on that, but anyone can take that. Dr. Lewis?
So just regarding for example, the implementation of rapid diagnostics, that is a much bigger thing. It's not just an easy click of the fingers. It depends on the kind of epidemiology of the area, the physician and consumer ways. So basically, from the side of rapid diagnostics, I would say that a lot of kind of research, possibly qualitative research, needs to go into really understanding the specificities of the area that you're targeting, and from there it needs to go to policymakers, and actually it's the policymakers who are going to have to really mandate the implementation of these diagnostics, as an example.
Yeah, thank you, Dr. Lois. I think I don't have any addition on that. We can move to our last question, and then we allow a session of question and answers from the audience. Dr. Sigei, reflecting on recent innovations, what lessons should the AMR community take forward to ensure technologies truly impact patient outcomes and public health in resource-limited settings?
Yeah, thanks for the question. And yeah, thank you. Quite a broad question. I'll try and do a piece of it, and I don't mind if my colleagues can add as well. I think at the end of the day, we must practice what we preach. I'll start by highlighting, the Global Action Plan on AMR, which I think is a document familiar to most of those on the call. And each country then has its own national action plan and so on. One of the key priorities is around innovations. We must increase, access to and also improve research on new innovations. How far have we done on that across our various countries? So there are those that have progressed. We have countries that are keen on local manufacturing, for example, and we've seen them, go quite far in that, whether it's on therapeutics or diagnostics. But we have others that are, not able to get there. And of course, pool procurement was something that was mentioned. That's something that I think we are still working on. It's a very good step, and we hopefully can increase how much we do of that. So I think our national action plans should be our compasses to see how far we are and what's missing. And especially the objective around new medicines, diagnostics, vaccines, and so on. But tied to that is the issue around behavioral. And I think this discussion comes up a lot in AMR forums. We can have all the policies and tools possibly available, but changing behavior, and I can speak as a clinician. changing the behavior of people actually testing before they treat is one of the hardest things globally, not just in Africa, but globally. And it's something that we need to, there are various, initiatives people have done, various studies ongoing on that, but changing behavior, making people actually say, before I treat, I must consider a test, whether it's a rapid test or a more advanced test, at least a test. And that's something that's really a headache for a lot of people. And then also we must think of, AMR has kind of segmented itself. It is a very important issue, but sometimes we forget it actually should fit into the broader health agenda where, patients with non-communicable diseases are the ones, some of them suffering the most from AMR. So sometimes we look at it in isolation and we really must see how to integrate. Obviously, I or many people don't have all the answers, but it's just a thought process that should lead us there. And then, of course, ultimately, we must put our leadership to task. We must demand a lot more from our leadership, especially around funding and prioritization. I like the word prioritization shared by the other speakers. At the end of the day, we must start somewhere. We must say, for example, in our hospital, we must be self-sufficient to treat the top 2, top 3 conditions that are affecting the country. So those are the kind of thoughts that I, came up with when I saw this question. And of course, I, open it up to the rest for their thoughts.
Thank you, Dr. Sige. I think
Okay, I just want to add to what Dr. Sige said. So I think the AMR community should actually focus more on, designing technologies that are affordable, that can be easily used and that can be adapted to resources limited settings. And we should, focus on the importance of, local context, like in development, like in technology development, to ensure that solutions that we are actually providing are compatible with what we have, like the structures we have in place and the trained specialists, like the medical laboratory technologists. And also we should focus more on building strong collaborations in the industry and the government, and also people in the research and academia world. We should work together, so that the innovations that we are actually getting can be effective. And also focus on sustainability, through funding. The government has to, we have to get out to fund all these diagnostics techniques that we're about to bring in. So through this and continuous support from the government it will really help to go a long way in improving the patient outcomes and the public health in general.
Yeah, thank you, Olabisi. So I think we can pick questions from the chat. So we have a comment from Murangoy. He says, thanks for the informative discussions. As highlighted on the funding constraints we are seeing, I would urge that this development be supported by economic modeling that will accelerate, that will generate evidence to show long-term impact in terms of monetary savings, but also health outcomes. I think that was a comment. Jacob, please go ahead and shoot your question.
Okay, sorry. I want to ask one more thing because, in the hospital setting, there's a rising of colicin resistance, and the last drug which is being used, especially to treat gram-positive bacteria. And also, you found that many treatment, especially in low middle income country, LMIC, they are usually based on empirical treatment rather than diagnostic treatment, which the culture result should come first with their disk being sensitive. But unfortunately, we found that doctors rely on empirical treatment rather than depending on culture result. How can you address that?
Thank you for your question. Would like to take this, Dr. Luis?
Would you mind? I heard that in LMICs that doctors often rely on empirical treatment, but would you mind just clarifying the question further?
Jacob?
Yes. Yes. Can I rephrase the
Can you
question? I was
rephrase your
asking.
question?
Okay, I was asking, in low middle income countries, in most patients can't afford full diagnostics for culture, in which they are doing culture, so they have to get maybe the full culture result. So it is a little bit very expensive. So in order to help the patient, especially in low middle income countries, doctors rely on what we call empirical treatment. How can you address that so as to combat AMR?
Thank you for your question.
So I think between us, we'll try to, I mean, first of all, I really understand how difficult it is in the African landscape when we do not have a strong diagnostic microbiology capacity. So I think on one hand, it's imperative. I think one of the previous speakers was saying we have to be demanding more. And I think some of the work that Africa CDC has done with the MAP project to really bring that into the spotlight and just show quite how undercapacitated the continent is in terms of its microbiology capacity. Some of the work that ASLM is doing, etc. This is where we really need to get a lot of momentum and be demanding that our countries, our laboratory systems, our microbiology systems are strengthened. Now that doesn't happen overnight, and it still leaves the clinician, if you like, working quite blindly, which is it's just not ideal, right? So without being able to have a patient-specific diagnosis, being able to fall back wherever possible onto some kind of surveillance system, which I understand is also needs to be strengthened. I mean, I think that's probably where the laboratory capacity I'm talking about has to go first, because to be able to introduce patient by patient diagnostics is also going to be costly. But so that the clinician has some idea about what are the most recent resistance patterns, etc. Because quite frankly, at the moment, with the way that many of you have to work blindly, it's really, really tough. So I wish I had a much more elegant answer to give you, but we have to build this laboratory capacity on the continent to at least give more surveillance. And yes, some of the innovation will help that. So whether it is, if you can culture, then how do you make sure that you get good readings or good understanding of those AST results? If you can't culture, what are we able to do with rapid tests to be able to get, at least, is it bacterial or is it viral? There's a long way to go. And I honestly believe that those are the building blocks that we need to be making a lot of noise about as African clinicians, African advocacy groups. And I'm hoping that Africa CDC's work with the MAP report is really starting to, if you're not measuring it, you don't know it's there. So they've at least opened up that challenge, if you like, or revealed the challenge. So sorry, I know that's not a direct question to a clinician who is struggling to then be able to access diagnostics, but this has to be an area of rapid advancement and direction of really funding, in my opinion.
I think, and just also to add on to that, the doctor mentioned that we have to be more demanding, and that's exactly what we need to do. I know that India has recently, I'm not sure if it was their national action plan or another document that they've just put together, where they mandate that every district hospital in India should have basic microbiology capabilities. So, having a national action plan in your country that actually really prioritizes diagnostics and microbiology capacity is really important. However, the next step is then the implementation of the National Action Plan, which comes along with its own challenges. But yes, we need to be more demanding.
Yeah, thank you, Cara and Dr. Ellis. I think that is well answered. And we are running out of time. We're actually winding up at the right moment. So I would like to take this opportunity to thank the panels and the speakers. Thank you very much. Just to give you one opportunity, at least, can you all, in one sentence, summarize today's topic? Then we can wrap up. One sentence. We start with Dr. Luis. She's ready. Then Carol, Sige, Olabisi, and myself and Namanya.
Sorry, I was writing my response. I would just like to say that it was very good to connect with other AMR activists around South Africa, as I think it's around Africa. Sorry, I think it's a really important thing that we all get to know each other and who's doing what around the continent, and work together, because we're not going to get anywhere without this, without working together.
Thank you, Carol.
I think my takeaway is that we don't always have to think of innovation as being out of reach. And yes, many of them may be, but let's think about those innovations that are being tailored to be able to use in low resource setting. Let's seek them out and let's see whether we can put some of these tools into play, and got to start somewhere. I think somebody said something. So we've got to start somewhere, and we can't just sit being completely overwhelmed. So thank you.
Yeah, thank you. Dr. Sige?
Yeah, I mean, first, thank you so much for the invitation. Really appreciated this session and also interacting with other AMR champions. To try and put it simply, let's try our best to practice what we preach. We have so many policies, guidelines, plans. We need to do our best to implement as much as we can. And borrowing a bit from the two previous speakers, we can start somewhere. If we're talking about diagnostics, we can have a network. Not all hospitals in the country need to have the high-end equipment. We can start working on sustainable referral models to ensure we get good results. If we're talking about academia, industry, and government coming together, we must discuss the elephant in the room, which is funding. So I think for me, let's work together, let's keep the collaboration, and we're always here to support. Thank you.
Thank you. Ms. Lawal?
All right, thank you so much for the invite. So for me, the key takeaway from today's webinar, I think, is that, for technologies to make a long-lasting impact on patient outcome and in public health in general, especially in LMIC, they must be very affordable, contextually relevant, and strongly supported by partnerships and sustainable models. So I think by doing this, to really help us a long way in the fight against antimicrobial resistance. Thank you.
Thank you. I think you've all summarized well. I don't want to add anything on top of that. So that is our takeaway for today. I would like to call it a day. Maybe if Yamanya has announcements for the next webinar. Yamanya?
Thank you so very much. And thank you to everyone who's been able to join us today. Our next webinar will be as scheduled the 23rd of September. It'll be a Tuesday at the same time. Looking forward to seeing you there. Thank you so much and bye-bye.